Okay, so today we'll talk about this other paper for this week that is related to a CRISPR-I screen that these folks run on astrocytes. But before we do, we'll finish up with a lingering question that we had from last week. From Monday. So on Monday we talked about this paper in which they see that this gene BIN1, which is a quick reminder, shows up second to ApoE as one of the genes that is most tightly associated with the risk of developing late Alzheimer's disease. Comes in many flavors, and one of them, the V1 variant, has the capacity to inhibit base 1 activity on APP by virtue of keeping APP out of endosomal vesicles that where APP would find base 1 and be cleaved by, right? So we saw at the end of Monday that the model they're working with is when cells do not have this other molecule, RIM3, being V1 exists in a closed conformation in which its SH3 domain is interacting with another one of its domains called CLAP, and because CLAP is needed for interaction with other things that would bring BIN1 to endosomes, the closed BIN1 stays out of endosomes. And APP molecules can go into the endosomes, merge with other endosomes that carry base 1, and when base 1 and APP are together, base 1 can go ahead and cleave APP, which will later be cleaved by gamma secretases and make A beta peptides. The reason I'm going kind of fast with all of this is because all of this we discussed on Monday. Yeah. So clathrin, Josh's question here is what makes clathrin so important? Clathrin, shown here, is a molecule that is associated with membranes and that works to, it's one of those molecules like ARF6 on the other type of endosome that works to sort things into endosomes. So when cells are starting to form endosomes or when they're starting endocytosis, they don't let just anything go into this endocytic pits, if you will. As the membrane starts invaginating, there's a bunch of things that are pulling from this region of the membrane from the inside, but not just anything can make it into these pits, right? So the same things that are pulling from the membrane from the inside may also be involved in interacting with things that are going to make it into these endocytic vesicles and end up in the endosomes. So the importance of clathrin is that clathrin is one of those, it's not the only one, but it's one of those proteins that quote unquote decides what makes it into an endosome or not. And in this case, according to the authors at least, clathrin is the molecule that would make APP go into these endosomes. But then clathrin is also the one thing that explains why when BIN1 V1 is in its open conformation, and the open conformation happens because RIN3 will now bind the SH3 domain of BIN1 V1, freeing the CLAP domain, and now CLAP can interact with clathrin. And in fact, CLAP stands for clathrin associated something something, so it's related to its ability to bind clathrin. So now CLAP binds clathrin. Clathrin can no longer, it's occupied by its binding with V1 and can no longer bind APP, and APP stays out of these endosomes. So molecularly speaking, the importance of clathrin is that it explains why when V1 is in an open conformation, APP stays out of the endosomes. Does that answer the question? Yeah. So we got a little ahead of ourselves, but the like I just said, the model that the authors propose is that when RIN3 and V1 are expressed at high levels in cells, RIN3 and V1 will bind and induce this open conformation of V1 that will compete for clathrin molecules, keeping APP outside of the endosomes. And when now these endosomes fuse together, what is brought together are base and something else, not APP, not its substrate. So base is no longer able to cleave its substrate because its substrate simply is not there, right? And that's how the authors propose the presence of bin1v1, which is say plus ring 3, can explain the inhibition of base 1, which would normally be involved in generating A beta oligos. But if you inhibit somehow base 1 activity, obviously that's going to lead to less formation of oligos, right? And this whole thing in turn explains why there's been this observed negative correlation between the expression of bin1v1 and Alzheimer's disease, right? The thinking is that for Alzheimer's disease to progress, in other words, for A beta oligos to continue accumulating to toxic levels and facilitating the formation of the A beta plaques, you need to keep base 1 active. And one of the ways to keep base 1 active, to keep producing A beta oligos, might be to take out bin1v1 out of the picture. So that might explain why in patients and in mouse models of Alzheimer's disease, as AD progresses, you see a decrease in V1 expression. On Monday, we sort of mentioned that the authors may have fallen a little short in their interpretation of the data when they said that RIN3 recruits V1 to endosomes, but not V9. There were all those images showing that when RIN3 and V1 are together, they end up in these endosomes, but when RIN3 is in the company of V9, nothing really happens. And what we said is that they fell short in their interpretation because they made it all about V1 and how V1 is recruited to the endosomes. But there was one aspect of those same data that they kind of failed to emphasize, and that is that just like V1 ends up in endosomes when RIN3 is present, it goes both ways. RIN3 ends up in endosomes when V1 is present, and this model explains it. That was sort of the core of the question that I asked for class participation on Monday. And not all of you, but many of you mentioned that just like RIN3 allows for V1 to go into endosomes, RIN3 is coming along with it into endosomes, and that's how RIN3 ends up in endosomes as well, only when V1 is expressed, but not when V9 is expressed. So that kind of changes a little bit the narrative that RIN3 brings V1 to the endosomes. No, they come together somewhere in the cytoplasm and then they go together into endosomes. Why? Because V1 has that clap domain that allows it to interact with clathrin, pushing out APP and leaving it out of those early endosomes. But the question that I wanted to ask of all of you, do you guys see any problems with this model? This model was primarily based on these biotinylation chase experiment in which they sort of painted all of the molecules that are on the surface of the cells with biotin. Then they let some time go by, and then they shave all of the biotin off the surface of the cell and essentially ask what has made it in, since we painted the surface of cells, what has made it in, what has been internalized. And in doing that experiment, they see that APP internalization is significantly delayed, right? That when normally it would take about 10 minutes for APP to go into inside of cells or internalized inside of the cells, if V1 and RIN3 are co-expressed in those cells, now it takes much longer for APP to make it in, to the point that it's no longer coexisting with base 1. So the idea here is that APP and base 1 go into cells at different times, and therefore they're not going to encounter one another, and therefore base 1 cannot cleave APP. That's the whole idea behind the model. Now I'm asking you guys, do you see a problem with this model? What you're saying is that base 1 should be with APP, should be internalized along with APP by 30 minutes, not before, because base 1 is what normally cleaves APP. When you're saying it should be there, you're almost assuming that because an enzyme has a substrate, the enzyme should follow the substrate everywhere it goes, right? But that's a very strong assumption. The point is that enzymes and substrates behave independently from one another, and what the authors are proposing is that because they're temporally separated from one another and base is going into endosomes earlier, by the time that APP finally makes it to the endosomes, base is no longer there. So therefore, APP cannot be cleaved. Would delaying the internalization of APP necessarily mean that APP will never encounter base? What Atosi is saying, so that everybody can hear, is that base 1 could come in again later, could be internalized later. And that's the whole point. In this experiment, they follow the base 1 or the APP that had been quote unquote painted by biotin, right? So they're following a very specific subpopulation of base 1 molecules and APP1 molecules, those that had been painted on the outside. Sure, those painted, those labeled molecules, base 1 went in earlier and APP came in a little later. But that doesn't necessarily imply the new base molecules are continuously coming in, are constantly being internalized. So if we assume that base 1 continues coming in, I have this canvas here, so hopefully we can pull this off. So the idea here is when you're looking at biotinylated base and APP, I can totally agree with what the data show. And what the data show is at 0 minutes, you'll probably have very little, if any, of both, right? Because these molecules have just been painted and it takes a while for molecules to be internalized. So what I'll do is I will say there is very little APP and very little base. Little a, little b. 10 minutes later, there is a lot of base and very little APP because its internalization is a little delayed. 30 minutes later, and notice that topographically, the newest vesicles are the ones that are going to show up later in the experiment. So you have to give them a while until they show up, but those are going to be the ones that are closest to the membrane. So 30 minutes later, you'll have very little of biotinylated base inside of these molecules because most of it has been internalized already, and now a lot of that delayed APP. So when it comes to biotinylated molecules, I totally agree. The biotinylated base never encounters the biotinylated APP. But if we make the reasonable assumption that both APP and base are continuously being internalized, whether they're biotinylated or not, then if we somehow looked at non-biotinylated APP and base, what we would see is that they're continuously being put in. Therefore, in these 30-minute late vesicles, there will be very little of biotinylated base, but there may be plenty of non-biotinylated base, right? So this model of delayed internalization, all it means is that they're not going to be simultaneously put together into these early endosomal vesicles from the moment that they were produced or the moment that they were synthesized. That's fine. So they're going into these endosomal vesicles a little later, or APP is going into endosomal vesicles a little later. But what all that means is APP will encounter newer base molecules that were more recently made or that are a little more fresh than the older base molecules. So in my opinion, this model of delayed internalization won't necessarily explain why on a steady state level, when you look at much longer time courses, we're talking a 20-minute delay here, and yet when they measure the cleavage of APP, they measure over 7 days, right? So what's 20 minutes on a 7-day scale? It's nothing, right? So in my humble opinion, it still doesn't explain. I don't think that they're not going into the endosomal vesicles at the same time explains it, because sure, APP is taking a little longer, but there will be new base molecules going into these first invaginations, we call them pits, and then the endosomal vesicles. So this is a continuous process that will continuously be filling up these endosomal vesicles with base, and APP may take a little longer to go there, but eventually we'll start getting into endosomes that are filled with base anyway. So let's imagine for a second that we don't shave the surface of the cells, right? So in my humble opinion, what that would have shown is that at 10 minutes there was base. So let me point to it because I'm pointing. So there would be base there, but no APP. At 30 minutes, there would be biotin-related APP. But you know what? There would also be a lot of biotin-related base. And from there on, they would have continued to be high level both. So the only time that you would have seen a difference is during those early 10 minutes. But if you were to continue this system of biotinylation and letting that go in, then you would have seen that it may be two different quote-unquote generations of molecules, but they will meet one another in these vesicles anyway. So the next paper that we'll discuss is CRISPRi screening in cultured human astrocytes uncovers distal enhancers controlling genes dysregulated in Alzheimer's disease. And in the introduction, one of the things that the authors point out is that genetic variants that are associated with complex traits. You guys remember those SNPs, for instance, that we mentioned sometimes in GWASs? People look for genetic variants present in people and their statistical association with conditions like Alzheimer's disease. Often, what happens is that those genetic variants that associate with disease are not in the protein coding region of genes or the promoter regions of genes. They often lie in enhancers, because enhancers do some of the fine-tuning of gene expression. And the thinking is when you start having genetic variants that affect coding sequence or promoter sequence, it's too much too suddenly, right? All of a sudden you're changing the amino acid sequence of a gene or the promoter such that now the gene may no longer be expressed, period. That's too much. On the other hand, if you make genetic changes to an enhancer, you may be changing that fine-tuning, and those changes in fine-tuning is what make explain a condition or disease, right? You're not killing the individual, you're just making them sick. Yeah. So they're interested in enhancers because genetic variants that are associated with complex traits, including diseases, often happen in them. How do we identify enhancers? With genes, it's relatively easy. You find open reading frames, meaning collections of nucleotides that would spell out codons that could be translated into proteins. You try and associate a classical basic promoter to them, maybe some splicing signals, and with all of that information, which you can extract from primary DNA sequence, nucleotide sequence, you can say, oh, I think there is a gene here. With enhancers, it's a little more difficult, right? Because there is rarely any strong signals to where an enhancer begins or ends, or what makes an enhancer. An enhancer is really a functional collection of binding sites for transcription factors that have the ability to direct the expression of genes that are far away from them, right? With that said, enhancers can be predicted based on not so much DNA sequence often, but often based on epigenetic marks, right? When you look at how the histones in different regions of DNA are decorated by specific post-translational modifications like acetylation patterns and methylation patterns, there have been some of these patterns that have been associated with enhancers, right? So sometimes you wouldn't necessarily look for specific DNA sequences or nucleotidic sequences in a genome. Instead, you look for the presence of these epigenetic marks. And when you find them, you say, oh, I think there is an enhancer here. The problem with that is that their functional validation and what target genes they actually regulate usually remains unknown. So there's a lot of effort into looking at epigenetic marks throughout genomes, using those marks or combinations of marks to say, I think there is an enhancer here. But the next step is what is usually not, is the one that is usually not taken because it's a little more labor intensive and costly. And that is to say, if I think this is an enhancer, what I'm going to do is I'm going to go in and perhaps mutate it or take it out, and then I'm going to try and identify the genes that this enhancer may be targeting, blah, 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 blah. All of that is a lot more work that a computer cannot do for you. So what they decided to do in this paper is combine enhancer predictions that were made based on these epigenetic marks that I was mentioning before. Anytime that, and we'll discuss that in a bit more detail in the next slide, but anytime that you see the word ENCODE associated with some genomic study, what that means is that it's part of a much larger effort, multicentric effort called ENCODE, that basically took it upon themselves to connect epigenetic marks to functional features of DNA, right? So these are this is the organization or the consortium of centers throughout the world that said, we're gonna do some experiments to see when and how we can map certain epigenetic marks to functional elements in DNA. So they combine enhancer predictions based on ENCODE with CRISPR-I, and we'll discuss what that is in a second, to identify about 160 new enhancer gene pairs, or EGPs. So that's what they're doing in this paper, okay? So let's take a look at how they did that. So first, once again, they're like, okay, we can have enhancer predictions, that's fine, but we'd like to test those enhancers. We'd like to experimentally validate them. So first, they needed to choose the enhancers. And to choose the enhancers, they combined two large data sets. They combined data that come from a technique called attack sequencing, and they combined the information that the ENCODE projects generated. And in this case, it was psych ENCODE because it was ENCODE applied to psychology or nervous systems. So we sort of already talked about what ENCODE is. It's using epigenetic marks to predict the enhancers. Does anyone by any chance know what attack sequencing means? Have you heard it before? I wouldn't blame you. It's a relatively new sequence, maybe about 10 years old. That sounds like, whoa, that's old, but in research scales, it's not that old. It's relatively new. Attack sequencing, if we were to put it, the simplest of terms, is a technique that allows you to determine what regions of DNA in a given sample are accessible, right? What regions of DNA are not engaged in tight chromatin conformations. And I'm not talking necessarily heterochromatin. Even some loose chromatin for attack sequencing would be relatively inaccessible. What we're talking about here is sort of quote-unquote naked DNA. Where in DNA do you see the DNA molecule pretty much by itself, right? So attack sequencing detects unprotected DNA regions. Or once again, accessible. So they took these two databases, one that looked at accessible regions in normal human astrocytes. That's what NHA stands for, NHA. So they looked at data that looked at DNA regions that are accessible in astrocytes. And then they looked at the psych ENCODE predicted enhancers. These was not necessarily made on astrocytes, it was made on entire nervous systems, but the idea is that psych ENCODE would have identified hypothetical enhancers in nervous systems, which of course would include astrocytes. And what they did is look at the overlap between the two. Any regions that encode said, I think this is an enhancer, and any region that attack sequences said, this area is accessible, they said, we're going to make them part of our list of candidates. So they had to be, the candidate enhancers had to be replicated in these two independent data sets. They included some others in some cases, but those were the main two. Then they also said, these putative enhancers or hypothetical enhancers that we would like to investigate should be in a TAD with more than one highly expressed gene. And once again, does anyone know what we mean by TAD? With itself. Excellent. Do you know what TAD stands for?
Topologically associated.
Excellent. Topologically associated domains. Topologically associated domains. Super briefly, when we think of DNA, we tend to think of DNA as this linear molecule, right? Long linear molecule. In some regions more compact, in some regions more loose and accessible, but aligned nonetheless, right? But reality is that cells, in addition to the whole compaction or less compaction of DNA into chromatin, there are these topologically associated domains. Now, the way that works is it turns out that throughout DNA, there are sequences that can be bound by specific groups of proteins, and these groups of proteins like to come together. So what you end up with is something that looks more like this. And what this does, and this would be the famous TAD, topologically associated domain, what this does, as Melita was in a way implying, is it allows for interactions between different regions of DNA with each other that are more likely than interactions between the same regions and something outside of their TAD. Right? So TADs are like, if you will, like neighborhoods or communities of genes within chromosomes that are more likely to interact with one another than with anything outside. So they said, we want our hypothetical enhancers to be accessible. That's what Attack 6 said. We'd like them to be decorated with epigenetic marks that are consistent with them being enhancers. That's what ENCODE said. We'd like these putative enhancers to be in a TAD where at least one gene is highly expressed. Because sometimes what happens is these TADs end up being silent regions of a chromosome where nothing is expressed. So what would be the point of going after enhancers that are in completely silent regions? Chances are you're not going to find anything in them. But if at least one gene is highly expressed, then there is the hope that the enhancer may be operational in those. With all of these filters, they ended up with 979 candidates. These are 979 regions of DNA that were accessible in astrocytes, marked with epigenetic marks consistent with them being enhancers, and in TADs that have at least one active gene. Let me... So then they said, we are going to use this 979 sequences or enhancers, putative enhancers, to conduct a CRISPR-I screen. Do you guys know what CRISPR-I is? I see you nodding. CRISPR interference. Excellent. So do you guys want to explain a little more what that means? So you both, right, it's CRISPR interference, that's what the acronym stands for, and you said it's silencing a gene without cutting it. So how does that work? Transcription. If we're talking CRISPR, we're past transcription translation. Yes. So it binds the promoter region to prevent transcription. Yes. You said the slicing part, you turn it off, right? Yes. Yeah. So it turns out, I'm glad that you guys were way more familiar with CRISPR-I than I anticipated. So for those of you who are not familiar with CRISPR-I, but may be familiar with CRISPR, and just so that we're all on the same page, CRISPR works by bringing this enzyme called Cas9 to an exquisitely specific location in DNA. I'm not even saying region, I'm talking location. Cas9 will be brought to one location and one location only of DNA by a thing called the guide RNA. So the guide RNA is a long RNA molecule, single-stranded RNA molecule that is preloaded onto Cas9, and it basically tells it where to go by means of the interaction between the guide RNA and DNA. Basically tells it exactly where to go. And the reason it does that is because the base pairing between the guide RNA and the region, the target DNA sequence is long. It's over, I think it's somewhere between 19 and 21 nucleotides in length. So you can achieve exquisite specificity in sequence when we're talking sequences of that length, right? A will happen 1 every 4 times in DNA. AC will happen 1 every 16 times in DNA. But now you do 19 nucleotides, and to find a specific 19mer of AC's G's and T's, it will happen 1 over 4 to the power of 19. So that's a very low chance that the guide RNA will take Cas9 just anywhere in DNA. It will take it to a very precise location. Now, in classical CRISPR, as Jacob was in a way implying, Cas9 will go ahead and cut the DNA. Right? So that's what those scissors are there to imply. That Cas9 is a nuclease. It can cut DNA molecules, both strands, right? And in classical CRISPR, what follows is we either let the cell repair that double strand break in DNA, which normally leads to deletions of small regions of DNA, because in trying to repair that break, the cell will often make some mistakes. So that leads to deletions. And that's how we're introducing a mutation with exquisite specificity. Or we can hijack the homologous recombination based DNA repair system in cells and tell it, oh, you're trying to fix that break? Here's the sequence that you need to repair that break. And in doing so, we're purposely inserting a new gene or a new sequence in that location. And that is what we normally refer to as DNA editing. Because we're not breaking DNA, we're actually editing it so that it now includes a sequence that we're interested in. Okay? So those are the two main uses of regular CRISPR. You're causing mutations at specific locations, or you're editing DNA at specific locations. Someone came in and said, what if we make this Cas9 non-catalytically active, right? What if we make it dead? And I mean that because when you use a dead version of Cas9 or d-Cas9, what happens is the whole complex will go to that super specific location that is dictated by the guide RNA, but it will stop there. Cas9 cannot cut DNA and it won't leave, it just gets stuck. It's trying to take that next step of cleaving and leaving, but it doesn't. So a dead Cas9 just gets stuck where it was brought to DNA by the guide RNA. And that is how, depending on where that dead Cas9 got stuck, you will affect the normal function of that location. If that location was a promoter, like Ale was suggesting, you may not allow for the binding of RNA polymerase. So you're essentially stopping transcription of the gene, because where the promoter is, where RNA polymerase would have normally bound to start transcription, there is this dead Cas9 taking its place, and it won't leave. If you do it somewhere in the middle of a protein coding gene, you may interrupt transcription of that gene. Transcription may have started, but then when RNA polymerase bumps into this thing that won't move, it interrupts transcription. The key here is that you can direct a dead Cas9 anywhere you want. And these people wanted to direct dead Cas9 to these hypothetical enhancers that they found bioinformatically. They identified 979 candidates, and they said, why don't we start sending dead Cas9 to each of them and see what happens to gene expression? Right? So that's the approach they took. They took those 979 candidates that they had identified bioinformatically, and they created a lentivirus library. What that means is they created a huge collection of lentiviruses that carried in them two things: a dead Cas9 sequence and a guide RNA. And those guide RNAs were designed to target different regions of each of those 979 candidate enhancers. So each lentivirus carried dead Cas9 and one guide RNA. But you have millions of different antiviruses which collectively carried guide RNAs, not more than one guide RNA for each of the enhancers that they wanted to test. Because they couldn't, these enhancers are long, right? So they couldn't say, well, I'm sure that if I direct Cas9 to this particular location, I will kill the enhancer. They didn't know that, so they designed several guide RNAs to target the same enhancer, just in case. In fact, they tell us here that what they did is they designed guide RNAs every 200 base pairs within each of those enhancers. Right? So you take any of those enhancers, they would design an RNA, a guide RNA here, then 200 base pairs later, and then another one 200 base pairs later, and so forth. Right? So if they had 980 enhancers, I'm rounding up there, and they designed several guide RNAs for each of them, let's say, in the tens of them for each, you do the math and you end up with, over 9,000 different guide RNAs that they designed. And they put each of those into a lentivirus, and then they created a library. So far, so good? Yeah? Then they take this collection of lentiviruses and they infect astrocytes with them. That's what's shown here. So, and these astrocytes now show up with those different colors to represent or to indicate that each of these astrocytes was not infected by one lentivirus. In fact, they were infected by several of them. They could see that because they could go into these lentiviruses and look for those guide RNA sequences that the lentivirus had introduced into these cells. And in most cases, they found that the astrocytes were incorporating a median of 7.4 guide RNAs into them. Some of them had fewer, half of them had fewer, half of them had more than 7.4 guide RNAs. So when you do these things in bulk, you achieve what is normally called in the jargon high coverage, right? So in this case, a given enhancer was not targeted in one astrocyte. It was targeted on an average of 330 different astrocytes, right? In those astrocytes, the other enhancers that were being co-targeted may have been different combinations in each of them. But the point is that from the point of view of a given candidate enhancer, that enhancer was on average targeted in 330 cells. Okay? And they're doing this with tens of thousands of cells. So then what they do is they let these they let the astrocytes express the guide RNAs and the Cas9, dead Cas9, let the dead Cas9 go to the enhancer where they're being targeted and stop or block the enhancer. So the idea here is that what you're doing is you're blocking the binding of the transcription factors that presumably need to bind to that enhancer for the enhancer to do its job, right? So just like in a promoter, dead Cas9 will stop RNA polymerase from binding and doing its thing. In an enhancer, dead Cas9 would be stopping transcription factors from coming and binding. You let that happen for several days, and then you take these astrocytes, break them up, but you do it on a single cell basis. So cell by cell, you send it to a separate well in this very fast machine, and you break it up. And from that single cell, you do RNA sequencing. And when you do RNA sequencing, you're going to be accomplishing two things. First, you're going to take a look at all of the RNAs that were present in those cells and in what amount. But also, you're going to be looking at what guide RNAs they had inherited or they had acquired through lentiviral infection. So you now know these two things. You know what enhancers were being targeted and what happens to the genes in those cells. So what you can do is you can group the cells, again, on an enhancer by enhancer basis. You can say for a particular guide RNA or a particularly targeted enhancer, I'm going to group the cells that did have that enhancer targeted and group the cells that didn't, and compare the expression of gene A, gene B, gene C, gene D, all of the genes in the genome. This is computationally massive, right? That's probably why this thing was done in a collaboration across many institutes, right? So this is a collaborative effort. So you guys understand the strategy here? You take a group of cells, you inhibit randomly enhancers in them, but then you have a way to cell by cell say, which enhancer did I inhibit here? What happened to the expression of genes? Second cell, which enhancers did I inhibit here? What happens to the expression of genes? So in time, hopefully, you'll be able to say, oh, look, in all of the cells where I inhibited enhancer 52, this happened to the expression of this gene. Check it out. All of the cells that I inhibited enhancer 138, this happened to the expression of gene Z and so forth. Right? So they came up with data that they displayed in this volcano plot, where on the x-axis, as usual, you have fold change. So that represents how much the change of a given gene, or how much the expression of a given gene changed between the cell with the enhancer inhibited versus not. And then on the y-axis, you have a sense for the statistical significance of the finding. No. Well, at the 330 was the mean number of cells or average number of cells where a given enhancer had been inhibited. So of the 979, pick a number between 1 and 979. 478. Enhancer 478 was inhibited in 330 cells on average. And in those 330 cells, I can ask the question, what happened to all of the genes when I inhibited enhancer 478? Does that make sense now? Yeah? Now, I got to say something that is important. I keep saying that they looked at all of the genes that were present in those cells. In reality, what they did is they concentrated on the genes that were present within 500,000 or 500 kb of that enhancer. If you're looking at what an enhancer does, it doesn't make sense to look at what happened to the expression of a gene that is in a different chromosome or millions of bases of base pairs away, right? We know that enhancers can work at a distance, but 500 kb is long enough from the enhancer, and they established that cutoff. So for enhancer 478, they said, all right, if I look from the enhancer 478 both ways, 500,000 base pairs in each direction, what genes do I see? Now I'm going to look at the expression of those genes, and I'm going to see if any of them change significantly. If any of them change significantly, then they establish what is called an EGP, enhancer gene pair, right? And each of these dots in this volcano plot here represents an EGP. And in an EGP, two things happen. There was a given fold change in the expression of the gene in that pair, and that change was significant or not, right? So the fold change is represented in the x-axis, the significance represented in the y. I have a question up here that we'll discuss in a second. So they said, all right, we're going to concentrate on the most statistically significant EGPs, right? We just want to make sure that these EGPs that we established are statistically significant. They set up a threshold, and all of those EGPs that made the cut represented 158 EGPs, or enhancer gene pairs, between 145 candidate enhancers and 116 target genes. Right? So from the 979 that they started with, they ended up with 145 that seemed to be doing something, to 116 genes. And enhancers and genes were paired up in 158 EGPs. And they're pretty confident that this thing worked, because they said, okay, we'll take a handful, this was 20 of them, we'll take 20 of these 158 enhancer gene pairs, and we'll test them separately. So we're not going to rely anymore on this single cell RNA sequencing data, because that's a lot of noise and deconvoluting of the data and whatnot. If these things are true, let's take 20 of these and let's test them separately. And they did that using, again, CRISPR-I. So again, they directed a dead Cas9 to where the hypothetical enhancer is. But instead of doing single cell sequencing, they just did nanostring sequencing, which is a different technology for measuring changes in RNA expression, right? And what they see is that there is a pretty tight correlate in the findings between the single cell RNA sequencing and the nanostring. Two separate experiments, two separate techniques showing the same thing. So now they're pretty confident that this here is not an artifact of the technique used. Is probably a true reflection of what happens to the biology of these cells. One question that I had for you for the class, real quick, is what do you think these dots here mean, right? So on the one hand, they tell us that they're colored orange, so they pass the significance threshold. But then on the other hand, were these genes differentially expressed, yes or no? What does a fold change of 0 mean? It didn't change at all, right? So how can you call something that was differentially expressed if it's not changing? So here's the key, and this is sort of the take-home message. It sounds completely counterintuitive. If you're telling me that something that you're looking at genes that change in expression, but then you identify for me a whole bunch of EGPs in which the fold change was 0, what are we talking about here, right? The thing here is that in approaches like this, in which you're dealing with tens of thousands of data points, sometimes you can achieve statistical significance with minute differences. So in this volcano plot, those dots seem to be at 0. But if you were to look at them, you would see that the fold change may have been 0.0001. Right? I'm like, okay, well, but a difference of 0.0001, how can that be statistically significant? When your sample size is in the thousands for your experimental and in the tens of thousands for your control, it may pass the test, right? So with these super large data sets, sometimes statistical significance can show up even if there is very little magnitude of change. So even though there may be very little biological effect, those differences, those minute differences are still deemed statistically significant. And they made a decision to accept even things that were that had a minimal fold change within their set of 158. We've seen other papers in which authors will draw, if you guys remember, 2 lines, and then they will pay attention to anything that happens to the left of the left line and to the right of the right one. Because the criteria may be, I don't want to pay attention to anything that is statistically significant, but biologically may be completely irrelevant, right? I want to see magnitude of change and statistical significance. So if that is the case, then you do that. These guys did it. Once they had identified these 158 EGPs, they started asking questions about what these EGPs look like, right? So we're going to go through these 4 questions, and I was hoping I could hear from you what you think the answers may have been. So first one is, how many genes did each enhancer regulate? Remember that I mentioned that anytime they home in on an enhancer, they would look 500,000 base pairs on either side, and then they would look at the expression of those genes, and any of those genes that change in expression significantly, even if it is a minute fold change, but significantly, they would establish an EGP pair, right? But an enhancer could be paired with more than one gene. So I'm asking you, how many genes do you think each enhancer regulated? 1, 2, 3, more than 3? So 1.5 meaning that if I had to draw this, there would be as many with 1 as those with 2, roughly, right? That between 1 and 2. And then 3 or 3 or more, it would be fewer. Everyone agrees. Does someone disagree? Does anyone think that, no, for sure, 3 or more? No? We're good with this? Okay. So somewhere between 1 and 2. Good. And let's flip it around. How many genes do you think, or how many enhancers regulated each gene? So a target gene could be targeted by more than one enhancer, right? So a target gene that is here could be targeted by an enhancer that is 200 kb that way, and another one that is 150 kb that way. Both would have been counted. So how many enhancers do you think, on average, targeted each gene? 2 again? Maybe 4? So you think that there'll be, I'm going to go with you, Mireya, so something like this, most 4, but then fewer 3, 1 or 2, and then fewer 5. Something like that? Okay. We're going to leave it at that for now. How far do you think the enhancers are from the genes they regulate? So here on the x-axis, we have the distance between the enhancer and the gene. What do you think? We always say, oh, enhancers can work from a distance, but how far would they normally be? All right, let me put it this way. Do you think that enhancers will typically work more on close genes than far away genes? So I see some yeses. So what you guys are saying is that most enhancers will work with close genes that are close, and then as we move away, the effect of enhancers on those target genes would drop. Gabi, I saw you nodding no, so I suppose that you think that maybe it's kind of flat, right? That no matter where the enhancer is, the chance that it would work on a gene, it's about the same. It doesn't matter if it's really close or really far. There'll be examples of both that are equally frequent because of the loops, right? Okay, leave it at that for now. Where do you think the targets are respected to other genes? So these guys came up with 4 scenarios. One is the enhancer targets the nearest gene. The enhancer targets a neighbor gene, but it's not the nearest. the enhancer skips a gene that is highly expressed, or the enhancer skips a gene that is silent. What do you think? I'm going to simplify this question. So which of these do you think is the most common scenario, Jacob? First or fourth? Okay. So what you're saying is, let's make that, so essentially first and fourth. Do I have an orange here? No. Let me see. Oops. I can't remember how I deploy this. Ugh. Anyway, sorry about that, but let's make it yellow. So first and fourth, and then the two other classes less. That's more or less what you're saying? Okay, any other ideas? The fourth one would be pretty common, more than the others? Yeah? So something like that. Okay. Yeah, come on. Trying to do this on the fly, no? Easy. Okay, cool. And last but not least, and this may come across as a really weird question, but do you think these enhancers were transcribed themselves? Do you guys think that enhancers are transcribed? Anyone for no? No, enhancers are not transcribed? Raise your hand. 4 hands. Anyone thinks, no, yeah, enhancers can be transcribed? Oh, now you're changing it.
I didn't need to say it.
So some yeses, that enhancers can be transcribed? Okay. No, yes. Well, we'll see. So this is what they actually found. In terms of how many genes each enhancer was shown to regulate, most of them regulated only one. And that is kind of counterintuitive, right? We're typically taught to believe or taught to think that enhancers are like operons in bacteria, that they're there in eukaryotes to coordinate the expression of several genes simultaneously. Yet when they looked in this particular cell type, in this experiment, most enhancers, most of them regulated only one gene. And most genes were regulated by only one enhancer. In terms of the distance, you guys got that right. In terms of the distance, most of the EGPs were between an enhancer and a gene that was within 50 kb in distance. As the distance got more than 100 kb, that dropped significantly. So typically enhancers weren't targeting genes that were past 100 kb. That may not have been all that surprising. This non-functional curve that you see here, they created by coming up with random enhancers and random genes, and it's sort of a sense for how far enhancers are from genes in the genome, and to show that it's not like an enhancer is never far away from a gene. They can be far away from a gene, it's just that they never regulate them. Okay? In terms of the distribution of types of targeting, the two main classes were the nearest gene and skipping a gene, but only if that gene was highly expressed. There were fewer cases of skipping a gene if that gene in the middle or that gene in between was not expressed. And if you will, surprisingly fewer of cases in which the enhancer would target a neighbor, but a neighbor that is a little further away. I suppose that you could make the argument that that goes well with this observation, that as you start moving away from enhancers, the chances that that enhancer will target the gene drop. So most enhancers will target nearby genes, and yeah, if there is a gene between the enhancer and the target, it better be expressed. If it's not expressed, it's not targeting the next one so much. And finally, and what I think is perhaps too many people the most surprising finding, because I bet that most of you didn't think that enhancers could be transcribed. That was something that was found about, again, a good maybe 15 years ago. Enhancers are transcribed. And in particular, among the functional enhancers that they tested, only around 30% of them were not transcribed. which is what we normally think of enhancers. Enhancers are not genes, they're not transcribed, right? But a good 30 to 40% of them were transcribed in one direction, and the rest were transcribed in both directions. So there is RNAs coming out of these enhancers that correspond to transcription of these sequences in both directions. You're like, hold on a second, I thought enhancers bound transcription factors, and those transcription factors facilitated the transcription of target genes. What are we doing transcribing enhancers? The reality is that now with newer techniques that allow us to see RNA molecules that live in cells very short times, we've been able to see, I say we, as in the collective we, we've been able to see that enhancers are often transcribed, in some cases in both directions. What do those RNAs do? We call them eRNAs for enhancer RNAs. What do they do? Not much. They get destroyed right away. That's why we had a hard time seeing them before. Why does this happen? The thinking is that when the enhancer brings the transcription factors to target and enhance the expression of their target genes, what they create is an environment in which all of these transcriptional activators are interacting with RNA polymerase to initiate the transcription of the target gene. But sometimes that RNA polymerase gets a little confused and starts transcribing the enhancer. Because remember that the whole gist of the enhancers is that they come in close physical proximity with the promoter. So the RNA polymerase is brought to that region, and the RNA polymerase thinks, oh, I should start transcribing from this core promoter onwards this protein coding gene or legit gene. But sometimes RNA polymerase gets confused and starts transcribing the enhancer, right? Everything is happening in the same physical location. That happens way more than we think, and that generates these eRNAs that don't do much, they get destroyed. It's a little wasteful, but it's something that happens. And it happened a lot with these functional enhancers, which gave them confidence that what the enhancers they were identifying were real, because that's something that we know happens with enhancers.